our next speaker is from Zagreb in Croatia. He's a professor of biochemistry, and he has been with us already at one of the symposia before. I would love to ask you, who in the room is familiar with the concept of aging clocks? Have you heard of that concept? Yeah, you are a knowledgeable audience, so several of you have heard of that. The concept of aging clocks was mainly created around the phenomenon of methylation, DNA methylation. So when we speak of aging clocks today, it's basically epigenetic clocks that are measured in most of the publications. Professor Gordan Lauc is going to present a completely different system which changes as we age in our body, which provides a fascinating opportunity to create a completely different clock. And that can be measured, and it is about sugars. So with not giving away the secrets, please, Gordon, share your fascinating research with us.
Thank you. So I will tell you something about glycans. Glycans as modifiable biomarkers and in the same time functional effectors of the age-related diseases. I know that many of you don't know much about glycans. We don't teach it in graduate schools, we don't teach it in university, but practically majority of proteins, at least those which developed after we became multicellular, are glycosylated. So they have a polypeptide component. These are the amino acids linked to polypeptide defined by a gene. This is a wisdom of the billions of years of evolution. And then they have a glycan component, which do not have a corresponding gene. Glycans are defined through a complex network of genes which work together, their polymorphisms, epigenetic regulation, environment, integrating into a chemical structure we call glycans, which is being added to the protein. And then it's a part of a protein function. An alternative glycosylation, adding different glycans to the same glycosylation site, is functional analogous to the coding mutation. So we know that coding mutation can kill you. You have a wrong amino acid, the protein doesn't work, you can die because of that. But adding a different glycan can have very similar consequences. And glycans are the ultimate layer of molecular complexity. But in the same time, they're the most neglected molecules of cellular communication. The protein you see revolving in the middle, that's the famous S-glycoprotein of the SARS-CoV-2 virus. Everything hairy on the surface is a glycan. There is no way we can understand the function of that protein without understanding the glycans. And the analogy I like to make are these two birds on the left. Normal bird and the bird who lost all the feathers due to disease. And you could be the best scientist in the world. You can have all the money in the world. You can plan the perfect experiment. You can execute it perfectly. But if you're trying to understand flight using this bird as a model, the closest to flight you could come would be this bird walking. Because bird without its feathers cannot fly. Protein without its glycans do not function normally. And one of the good examples are immunoglobulins. So the so-called FAB domain, the fragment binding to the antigen, it's a polypeptide. But what will happen after this antibody binds to the antigen is actually defined by a glycan in the Fc domain. So antibody can be pro-inflammatory, it can cause the activation of the immune system and the killing of a target, but it can also suppress inflammation. And this is determined by alternative glycosylation, adding different glycans to the same antibody. Before I go on, a word of caution. Term glycation is way more familiar to all of you, but glycation has nothing to do with glycosylation. Glycation is a chemical reaction, it's a random damage, it's taking a shotgun and firing at the protein. We usually quantify it as HbA1c. It does correlate with aging, but it has nothing to do with glycosylation. So please do not think about anything you know about glycation when you listen to my presentation. One thing which is very peculiar or specific for glycans is that they're highly modifiable, they're highly variable. For example, we analyzed a cohort of several hundred mouse strains which were defined by the Professor Grant Morahan in Western Australia, where he started from the 8 founder strains, crossed them through 3 generations, and then inbred them into strains. More or less, he made clones of cousins. And some of these clones became completely different from the rest of the population. And there were no mutations in these 3 generations. So these differences were not caused by genetic variation. They were caused by reshuffling the allelic variants, which made mice completely different. So it's a form of rapid evolution creating stable and heritable differences in protein structure and function. And we also have it in the human population. So one of the reasons why we are all not the same is because we glycosylate proteins in a different way. Even blood groups are chemically glycans. What you see on the right-hand side is 6 random people, different glycan, different structure, attached to IgG. People are different. And these differences have genetic, epigenetic, and environmental factors which affect them. And one of the things which changed through time, as we are aging, is protein glycosylation. And this is something we published over 10 years ago. Actually, on the very same day where Steve Horvath published his epigenetic clock, we published our glycan clock. And then editors published an editorial asking, are glycans the holy grail of biomarkers of aging? I believe they were right, and I hope you will believe it too after I finish my presentation. We analyzed over 200,000 people. We looked into glycans in different cohorts, genetic, epigenetic, clinical. And in many diseases, nearly all of them which we studied, we see that people with the disease change glycans in the same way as glycans change with aging. So healthy, old people have their glycans similar to the young people with different diseases. Diabetes, lupus, Crohn's disease, IBD, many different diseases. And actually what we think we are seeing here, we are seeing the glycans as a tool to modulate low-grade chronic inflammation, and they're part of the disease process. And the actual disease name will depend on a symptom or location, on genetics, on epigenetics of an individual. But the process itself is heavily dominated by changes in glycosylation. Because the glycans on IgG are functional effectors which have multiple roles in balancing inflammation. And when we are young, we mostly have structures which look like this, and they're actively suppressing inflammation. As we are getting older, more and more of our immunoglobulins will have structures like this, which are actually promoting inflammation and putting our organism under stress, which eventually breaks down in one of the chronic diseases which we know. So kind of a big picture is, mostly we see our patients here when something breaks down. So something is not functioning, and then you go to hospital and ask for help. And usually you get the name of a disease, you have some chronic disease, and you have it for your life. That's this lifespan without health, which we heard about. But this is not happening at a given moment. This is a process which takes over a decade to happen. And molecular changes are identifiable long before we actually got diagnosed with the disease. And this is something what we see with glycans. We see glycans changing up to a decade before disease. We know they contribute to disease development. And if you find a way to intervene here, to change them, to improve them, before people actually get ill, we can postpone diseases for this extra decade of healthy life we want to have, and either do not develop disease at all or develop it very late in our life. We did many prospective studies with samples collected a long time ago, and then people develop diseases later. And we clearly showed that glycans change up to a decade before people develop diabetes, even before people develop insulin resistance. Also, glycans are probably the best predictors of future heart attack and stroke. And these are all very large populations, a large cohort published in good journals. So this is something we really can believe in the data. Also, we see glycans predicting complications of diabetes. We see glycans predicting hypertension, inflammatory diseases, even severe COVID. Based on glycans, we could predict who will get severe or easy form of COVID. And also, glycans are one of the first indicators that somebody is entering the perimenopause. And based on all that knowledge, we will develop this glycan clock of biological aging, which is chemical structures, glycan structures, which we can reliably quantify, which are the functional effectors of inflammation, and they can be modified both by lifestyle and pharmacotherapy. And here, of course, I have to disclose I'm the named inventor, I'm a co-founder of the Glycan Age company, so I'm here definitely conflicted. So I will do my best to show you the data, and you make your own conclusions based on the data. For example, a large study done in Qatar analyzed over 6,000 different phenotypes. They measured more or less anything that can be measured in a human. And when they looked for correlation with aging, 9 out of the 20 strongest correlations were actually glycans, immunoglobulin glycans. The other 11 were mostly sex hormones. So glycans are clearly strongly correlated with changes that happen with aging. We are trying to understand how and why this happens. So in thousands and thousands of people which we analyzed, many of them also had the genetic information. So we did so-called GWAS study, genome-wide association studies, to see which genes regulate glycosylation. We mapped over 40 genes which regulate IgG glycosylation, and some of these genes are known risk factors for a number of diseases where we see glycans altered. We also estimated heritability, so approximately 40% of glycan age is heritable, defined by our genetic information, while the 60% is affected by our decisions, by our lifestyle. The way we live is determining how fast our glycans are changing from the inflammation suppressing to inflammation promoting glycans. And this is something we can also change. So these disease predicting glycans can be changed with the lifestyle and pharmacological interventions. We also published a number of papers showing that physical activity can help, dietary interventions, hormone replacement, anti-inflammatory biologicals, and interestingly, even the exosomes from this was a study done in rats on a porcine plasma, but they were extremely effective in reducing both glycan and epigenetic age. One of the best examples of diseases we have, and I will show it here, is the cardiovascular diseases, actually cardiovascular events, so whether somebody is going to have heart attack or stroke. So our first indication that glycans associated with cardiovascular diseases risk came from a cross-sectional study of over 3,500 people in UK. We showed there is an association. Then we looked into the prospective cohorts. We got samples from the German part of the EPIC cohort with the 27,000 people collected 30 years ago, followed up clinically, and we knew who did and who did not develop cardiovascular events. And we showed that the glycans in men are as predictive as entire AHA score, in women even more predictive. The single glycan resulted in the hazard ratio of 0.7 per standard deviation in women. Just one study, we had to replicate it. We did the replication and got nearly exactly the same hazard ratio for women, also different glycans in men and women. And here we developed this cardio age test. Again, I'm a named inventor. This is not commercially available yet, but it's available for clinical collaborations. Did additional replication together with colleagues on Harvard. This was just published a couple of weeks ago in Circulation Research. Again, nearly exactly the same hazard ratio as before. So clearly, immunoglobulin glycans predict future heart attack and stroke up to 10 years before it happens. And interestingly, this can maybe even hacked. At least in mice, if you feed them with a dietary supplement called MANAC and acetylmanosamine, immunoglobulin glycans can change, and actually it can protect obese mice from developing hypertension. We don't know whether this works in human, but we know that the weight loss works in human. For example, bariatric surgery is very efficient. We had a patient improve 37 glycan years in 6 months. Also, the regular types of weight loss without surgery help. And we had 1,000 people on a low-calorie diet for 2 months, 800 calories. That was a torture, but their immunoglobulin glycans improved. Then they put them on 5 different maintenance diets. And in each of the 5 diets, some people got better, some people got worse. Because we are all different. There is no one-size-fits-all approach to healthy diet. There is no universal healthy diet. We have to find a healthy diet which works exactly for us. One of the good things which came from our research is that if you lose extra weight, independently whether you're in your 30s, 40s, and 50s, your glycan age will start improving. So it's never too late to lose the extra weight. Physical activity also helps, but can also do harm. So we were misled into believing that you can sweat out the bad diet. You cannot. So too much physical activity is detrimental. And when we compared active people with controls, which for Croatia would be obese, sedentary individual, active peoples were younger, but professional athletes were actually worse than the controls and the active people. If middle-aged people started to exercise, even up to 9 months, we would see more pro-inflammatory processes going on, because usually we overtrain people. So when we decide to change something, we do it too much, and then we do ourselves damage. One good information here is that glycan, which was cardioprotective, which was predicting heart attack and stroke, it did improve in all the groups. So maybe you get more inflamed, but even with physical activity, even if you're exercising too much, you're probably protecting your cardiovascular system, which we can quantify using glycans. Another set of glycans is the total plasma glycan, which we looked. Previously, I was talking about IgG glycans. Total plasma is taking all the proteins, cutting off the glycans, and see what they do. And they predict diabetes. They predict diabetes and insulin resistance up to 10 years before they are diagnosed. And this is changing gradually. This is 10 years before diagnosis, 7, 4 at diagnosis. So glycans change as we are becoming closer to diabetes. And if we do a lifestyle intervention, which is known to be healthy, we see these glycans changing in the opposite direction. So actually, you can revert this damage which has been done by unhealthy lifestyle, and you can quantify it on an individual level. So you can actually show your individual patient, yes, this what I'm giving to you is actually working. And this is very important for motivation, because one of the key problems in the longevity field is you do an intervention and then say, yes, you will live 10 years longer. Okay, who knows whether I will or I will not. But if you can quantify something and show your patient this really works, this is really helping. Another important aspect is menopause. When we looked at the population cohorts, blue are men, red are women. Women are protected for this, from this low-grade systemic inflammation for the big part of their reproductive life. And then you see a very rapid acceleration in all the cohorts that we looked around the age of perimenopause. And when you look at individual level, and here we tracked 2,000 twins through 15 years in 3 time points, you see a rapid acceleration at the age of perimenopause. And actually this happens as one of the first symptoms, because the problem with the perimenopause is that quantifying hormones doesn't really help much, because hormones get crazy. They change within a day, they change between days. So you have to make multiple measurements to actually get reliable data about the hormone levels. IgG glycans are doing that for us, because they're under control of estrogen. They have a half-life of a couple of weeks. So you take one time point of IgG glycan and you get an integration of the weeks of estrogen concentration. And then if you do a hormone optimization, these things usually get better, and you can also tune it to the individual patient. Again, I'm conflicted. I'm inventor on a patent. But for example, if we looked at the consortium of biomarkers of aging, they clearly identified, they published a paper in Cell about biomarkers of aging. They said glycan age associated with multiple diseases. It's actually the only commercially available test used to monitor lifestyle interventions. And I do hope that I have convinced you that glycans are the holy grail for biomarkers of aging, because they integrate genetic, epigenetic, and environmental factors. And we all know that we have to do preventive maintenance on our cars. If we would not, driving would be very deadly, but we don't do it to our bodies. And all our research is funded by many grants. We got over 40 million euros of grants, and I have to thank you to our funders who enable us to do the research. Thank you.